Alright guys, let's look at DNA replication. So in this lecture we're going to talk about how cells make more copies of their DNA. And we will finish off by talking about what happens when that process goes wrong. Specifically, mutations is what happens when that process goes wrong. So just a reminder before we get started. This right here is if you recall from the very beginning of the quarter, we called this the central dogma of molecular biology. Altogether, besides being called the central dogma, this is also called gene expression. So this is also called gene expression. Now, if we just look at one part of gene expression from DNA to RNA, remember that's called transcription, and from RNA to protein that's called translation. We are looking at neither of those today. Instead, what we are looking at is DNA replication. So how we make more DNA out of our pre-existing DNA. One small thing to note, we'll actually see a little bit of this later on. You can, in some very rare cases, actually do what's called reverse transcription, where you can take RNA and turn it back into DNA. So as humans, we don't really do this, but many viruses do. And they're actually called retroviruses because they do transcription backwards. Retro means backwards. As far as we know, there's nothing that's capable of going from protein to RNA, though. There's no living thing capable of that. So, we're really going to focus on DNA replication here, and just a reminder, DNA replication occurs during S phase of the cell cycle. If you recall, we have G1 followed by S, then G2, and then we actually divide during mitosis or meiosis. Well, we're going to look at what happens during S phase. So DNA replication only happens during S phase. In contrast, when we look at transcription and translation, assuming that my PowerPoint cooperates here, there we go. Transcription and translation take place during the entire cell's life. Your cell is always doing transcription and translation. If we stopped transcription and translation, our cells would die pretty fast. DNA replication, though, only happens during S phase. And remember, not everybody is actively dividing, right? Not every cell is actively growing. In fact, most of our cells are in G0. So DNA replication doesn't take place all the time, and it only takes place in certain cell types, certain actively growing cells. Alright, this is also a little bit of a reminder too. Recall that when we look at DNA inside the nucleus, most of the time, and here's the nucleus right here, I'm outlining the nucleus in red. Most of the time our DNA is what we call decondensed. It's all spread out. It's not tightly compacted. So DNA is decondensed during interphase. Remember, interphase is G1S and G2. The only time it really starts packing down is during M phase. That's where we see the condensed chromosomes start to appear, specifically during prophase of M phase. And this tends to be a common misconception amongst students, as they think that during DNA replication we start condensing. That is not the case. The entire time during DNA replication, your nucleus is going to look something like this on the left instead. And keep in mind that when we see a nice, neat karyotype like this, remember a karyotype is all of those chromosomes artificially arrayed, that is also taken either right before mitosis or right after mitosis. Alright, so I want to give you a quick reminder of the structure of DNA. I don't know if you remember way back when, but at the beginning of the quarter we said that DNA has a really unique structure, and it means that we can only read it in one way, and we can only write it in one way. Just kind of like when you write English, right? You only write from left to right. You only read from left to right. Well, DNA kind of works the same way. We can only read and write in a certain direction, and it's all based on its structure. So a quick reminder about its structure. Remember the backbone right here in red, it's called the sugar phosphate backbone. It's made of deoxyribose, the sugar, and phosphate. Alright. If you're thinking ribose, that's RNA. We're not looking at RNA replication here. In fact, humans don't really do RNA replication, so we are never going to worry about it. And our DNA, our nucleotides have three components. Remember, nucleotides are the monomer of DNA. Nucleotides have three components: the sugar, the phosphate, and together those are the sugar-phosphate backbone. And then we also have the nitrogenous base. And in DNA, there's 4 nitrogenous bases: A, T, C, and G. A pairs to T, C pairs to G. And these little, the reason why I do 2 here and 3 here is because there's 2 hydrogen bonds that hold together A and T, and 3 hydrogen bonds that hold together C and G. So these base pairs, when they pair up like this, they're called complementary. Notice the spelling here with an E. Right, these complementary base pairs are held together by hydrogen bonding. And remember, in order to get that hydrogen bonding all aligned at the right angle, if one DNA strand is, we say it's running, right? If one DNA strand is running 5' to 3', the other one has to go the opposite direction. 5' matches up with 3', and we go from 5 to 3, and we say this is anti-parallel. Right? This is anti-parallel. If it were parallel, there'd be a 5 up here and a 3 down here, but it's not parallel. And again, this is due to how the hydrogen bonds have to match up. Right? We have to have the right angles right here in order to get the nitrogenous bases to properly hydrogen bond, and in order to do that, we have to be anti-parallel. And DNA, remember, it twists around itself, so it exists as this double helix. Double because there's 2 sugar-phosphate backbones. Alright, so this is what happens to DNA during DNA replication. We start off with a single line of DNA. Alright, we start off with a single line of DNA. Notice that in that line of DNA, we have a couple sequences, the centromere. Right, remember the centromere is what we grab onto during mitosis to pull the chromosomes apart. And a chromosome is simply a single, really, really long line of DNA all compacted and squished up. In orange, we have what are called the replication origins. You can probably guess just based on the name what those do. That's where we start DNA replication. And then later we'll see what these are in blue, the telomeres. So, this is your cell's DNA during G1. During S phase, you start to replicate DNA beginning at the replication origin, right? Go figure. And what'll happen is we start replicating DNA right at that replication origin, and these little bubbles right here, they'll move outwards in either direction until you have two copies, two hopefully identical copies of that original DNA molecule. So at this point we are at G2. Now at the end of G2 we condense our DNA, and that's when we start seeing those condensed chromosomes. That's when our spindles will start attaching to these centromeres. We divide during anaphase and we end up back with our original single chromosome copy. Alright, so that's our overview of when this happens. So mechanically speaking, DNA replication undergoes what's called semi-conservative replication. Semi-conservative. We're not referring to the political stance here. You're not like halfway Republican, right? Semi-conservative just means preserving half of the old molecule and introducing a half new molecule. So what does that mean? Here's where we start out with the old, what's called parent molecule. This is what we see, by the way, during G1. This is what we saw in the previous images, G1. As we approach DNA replication, we'll break the hydrogen bonds between the nitrogenous bases. We'll break those hydrogen bonds, and we'll separate the two strands, the two sides of that parent molecule. Now, if you know one side, you know the other side, right? If you see an A, you know automatically, even without having to look at what was on the other side, you know that there had to have been a T there. If you see a C, there had to have been a G there. If there's T, A, right? A, T. I had a student, it's kind of hard to remember T bonds to A and C bonds to G sometimes. Right? You have to think about it for a second. I had a student tell me that the way she remembered it, and I will warn you, this is really rude. Okay, so if you have kids listening, you may want to plug their ears. T and A go together, right? Tits and ass. There you go. You're never going to forget it, huh? So you can unplug your kids' ears. Alright. So because we know one side, we can very easily match up, using enzymes, a new, what's called daughter strand. Daughter strand's in light blue. Alright, so a daughter strand, or the end result of this will consist of one half of the parent molecule and one half of a new DNA molecule that you've made. So we say that it's semi-halfway conservative. So right here, this is what we get during G2 of the cell cycle. This happened during S phase of the cell cycle. Alright, so again, zooming in a little bit deeper. Alright, we're taking it at the top level and then going deeper and deeper and deeper and deeper. We're looking at the same thing here. Here's our parent strand. Here's the new daughter strand that we are laying down. Okay, same color scheme, dark blue and light blue. Now we're saying exactly how we add on new nucleotides. In this, I really like this picture because you can actually see the three components of a nucleotide. Right? You can see the nitrogenous base. In this example, it's G. We can see the 5-carbon sugar, deoxyribose, and that's the phosphate group right there. So, keep doing this. After we have unzipped our parent DNA molecule, it's single-stranded, and we can come in and match up daughter nucleotides. And here's how we power this process, and here's the enzyme that does this. Well, the enzyme that does this, look at him, DNA polymerase. DNA polymerase is the one that makes a polymer of DNA. Go figure, right? Somebody was really proud of themselves when they named that enzyme. Now, how we actually power this process, because remember, we do have to power it. It's creating, so we have to input energy. This is an endergonic process. We actually power it by using, well, remember ATP? Right? We actually don't use ATP. We use a DNA-based version of ATP called DATP. This is not something you need to know, at least not at this level, but you may in future classes. So, if you remember, ATP stood for adenosine triphosphate, and ATP had, of course, an A nitrogenous base, a ribose, notice ribose, and then 3 phosphate groups. So here we are actually going to replace the ribose with deoxyribose instead to make a DNA version. And so that's what the D right here stands for, deoxyribose. So a DATP can be hydrolyzed into DADP, and that can be further hydrolyzed into DAMP, just like regular ATP can be hydrolyzed into ADP and AMP. We can also make these nucleotide triphosphates out of all of our other nucleotides too. We can make DCTP, which will become eventually DCDP, DCMP, right? We're just using C instead of A for a nitrogenous base there. We can make DGTP, there we go. It's a tongue twister, alright. We can make what's my last one? TTP. Our cells make all of these without too much problem. They're just constantly making these nucleotides whenever as they are approaching S phase. So, what we see here is a deoxyribose. It has to be deoxyribose since we're making DNA. Look at the nitrogenous base, T. There's 3 phosphate groups, so phosphate. We have a DTTP right here. And what we're going to do is we're going to match that T, nitrogenous base, to the A on the parent strand. And in order to create the bond between this T right here and this other nucleotide, the G, we're going to chop off these last two phosphate groups. So I know previously we just chopped off one. We went from ATP to ADP, triphosphate to diphosphate. Here we're going to be even more extreme. We're going to chop off two. We're going to go from TTP to DTMP, monophosphate. And of course, chopping off those phosphates, here we see those phosphates that are released. Chopping off those phosphates does release plenty of energy, and DNA polymerase takes advantage of that energy released to create the bond. That bond right there. Actually, let me be very clear. There we go. That bond between the T and the previous G. So nucleotides power their own polymerization with the help of DNA polymerase. Let me say that one more time. Your cells produce nucleotides, DATP, DTTP, DCTP, DGTP, that power their own polymerization. This is our same exact energy coupling reactions that we have seen before, just in a different context. Okay, so that's the very, very nitty-gritty of how this happens chemically. Now, of course, it's not just that simple as unzipping, sticking in some nucleotides, and saying we're done, right? It's never that simple. So the process of actually doing this is a little bit more complicated. Remember that we said we can only read DNA in one direction and write DNA in another direction? Well, that's mostly why. So let's take a look at that process. First, we have to look at how this process gets started, right? In order to get replication started, we have to form what's called a replication bubble. Let me show you what a replication bubble looks like. I'll come back to this slide. So here's a long eukaryotic chromosome. Remember, our chromosomes are just really, really long lines of DNA that are highly compacted during mitosis. Okay? Let me actually erase that. Oop. Keep doing that, man. There we go. So, at several points in our chromosome, we have special sequences called replication origins. Remember, we saw that before. They're also called origins of replication, whatever you want to call them, replication origins, origin of replication. It's the exact same damn thing, okay? So we start opening up our DNA molecule at the replication origin. We rip apart those nitrogenous bases from each other and break hydrogen bonds. When we do that, we open up this little bubble. It literally looks like a little bubble, and it's called the replication bubble. Alright, the replication bubble. At either side, right here, where the DNA forks, as it enters that replication bubble, that's called a replication fork. Replication fork. Replication bubble. What do you eat DNA spaghetti with? A replication fork. You better not steal that for your exam extra credit. Okay. So in order to do this, we actually have to start unwinding DNA, and of course it's an enzyme that does this, both in prokaryotes and in eukaryotes. It's a very, very similar enzyme. Alright. Once we have unwound, we're going to proceed bidirectionally. Bidirectionally means in both directions, and we also saw this before too. Let me erase my arrows so we don't get confused. Come on, arrows, there we go. So when we say we proceed bidirectionally, we're going this way and this way. The origin of replication was right there. So we're proceeding outwards from the origin of replication. Okay. And eventually these replication bubbles will meet right in the middle there and merge. What exactly happens when they merge, we don't really know. We just know that it happens and we're grateful for it because it means we get to stay alive. So you'll notice that eukaryotes have multiple origins of replication, and that's because we have a lot more DNA than prokaryotes. Prokaryotes have only one origin of replication. But on average, we have way more, so it takes a lot longer to replicate our DNA. If we had just one origin of replication, we started there and we went outwards, it would take way too long to replicate our DNA. I forget the exact numbers, but I think it's something like 3 months to replicate all of our DNA. So we start at multiple locations and we divide up the labor to speed the entire process up. Bacteria don't have to worry too much about that because their genome is so much smaller. So they have one origin of replication, they'll open up one replication bubble, they'll only have 2 replication forks, and they'll go around in either direction. This is just a really cool picture. This is actual DNA being replicated, and you can see that replication bubble. Okay, so we looked at origin of replication. We know from the origin of replication it spreads outwards in either direction. We have bidirectional replication. Bacteria do too. But we need those multiple origins of replication. And a replication fork is that Y-shaped region where we're proceeding forward. I'm at the point where the word replication doesn't seem like a real word anymore. Replication, 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 replication. Okay. So, as we just said before, we need to actually unwind that DNA and break the hydrogen bonds. It's hard to do that, right? So we recruit the help of an enzyme named helicase. So helicase will unzip DNA by breaking the hydrogen bonds between the parent strands, all right, to separate them. And helicase, I think he goes by a couple different names. I think he also goes by DNA gyrase. We call him helicase in eukaryotes, and I think we call him DNA gyrase in bacteria, but they both do the same thing. Now, helicase's activity kind of creates a bit of an issue. You ever take like one of those old-time twisted telephone cords, right? The coiled telephone cords when you were a kid and you tried to straighten out the bottom? And the top became super twisted while you straightened out the bottom? That's actually exactly what happens with DNA. Remember, DNA is twisted. So as we unzip the strands, we straighten it out. And as we straighten it out, we introduce some super coiling, some really, really highly coiled bits further up. And DNA is a pretty delicate molecule. If we do that with a little too hard, we're actually just going to break the DNA molecule right here where it's supercoiled. So we have to recruit another enzyme called topoisomerase. He will relieve that torsion tension caused by the helicase. And if you look at the name of this guy, his name actually makes sense. Topo refers to the surface of things. So you guys, have you ever looked at a topology map? Well, topology map tells you about elevation, tells you about the surface of the Earth. Isomerase, right? We know what that one is. It means rearranging. So a topoisomerase will rearrange the surface of DNA. And how it does that is it actually cuts DNA, physically cuts it, then allows it to untwist and then rejoins it. It's pretty cool. So we have an entire class of anti-cancer drugs called irinotecan, and irinotecan is just one of these drugs. Another example is something called topotecan, obviously named after topoisomerase. These are chemotherapies, and they inhibit topoisomerase. So the cells that are doing DNA replication the most are going to be cancer cells, right? So if you inhibit topoisomerase, every time they go to do DNA replication, their DNA is going to get super twisted because they don't have the ability to untwist it, and it's going to break. So irinotecan and topotecan are both work quite well as chemotherapies. Okay, so once we've unwound our DNA, this is where it gets a little hairy, okay? Now we have to make our daughter strands. And let me actually show you I have a blank slide over here. Let me draw this for us. So, just to give us a basis of comparison, I'm going to use the same color scheme. Here is my, here is my screwed up slide, there we go. Here's my parent strand. Okay. Oh, those are crooked. There we go. Okay. Are you going to cooperate with me? A little better. I swear I'm not drunk. Alright. So here's my parent DNA molecule. I'm going to open up at the origin of replication. I'm only going to show one origin of replication because, well, I'm going to be lazy, okay? And what we are looking at here is adding our daughter strands in light blue. What I'm going to do is I'm going to zoom in. I'm just going to focus on one of these replication forks. Okay, so I'm going to draw one of these replication forks a little bit bigger. Let's do it like this. I'm just drawing the backbone. I'm not drawing any of the nitrogenous bases. This is just to kind of keep the diagram a little cleaner. Okay. So, when I make my daughter strand, okay, let's just look at focus on the top one for now. When I make my daughter strand, I'm going to match up what I see on the parent strand. So if I see an A up here, T, C, G, G, just as an example, I'm pulling this from my rear end. These nucleotides don't have any significance, I just want to show you how they match up. I'm going to put an T on the daughter strand, A, G, C, C. Okay? So far so good. Now remember that whole thing about only being able to read DNA and write DNA in one direction? Well, as it turns out, let me remind you of let me tell you about these rules, rather. Not remind you, this is the first time we've seen them. So, we can only read DNA, our enzymes, rather, can only read DNA from 3' to 5'. Our enzymes can only create new DNA, or write DNA, from 5' to 3'. So what does that mean? Well, let me add labels on here, and hopefully it'll become clear after that. So if I'm creating let's look at my top daughter strand. Alright, I'm going to erase the nucleotides just to keep this image clean, because there's going to be a lot going on here in a few moments. So if I can only read DNA in one direction and write DNA in one direction, let me give you some directions. So remember, we're actually looking at this side right here. My replication bubble and my replication fork are going to move that way. So my daughter strand is also going to be moving to the left. So let me add some arrows here. If we're reading from 3 to 5, that means that 5 has to go right here, 3 has to go right here, because I'm going from right to left on this image. So I have to read. Oop. It helps if I actually put the right labels, don't I? It doesn't at 3 to 5. There we go. Sorry about that. Okay. So if I have 5 over here, that means I have to have 3 down here. And if I have 3 right here, that means I have to have 5 right here. Ignore all that. Okay, I'm just fighting with this here. So if I have 3 up here now, again, just using my anti-parallel base pair rulings, that means 5 has to go right here. That means 3 goes down here. Alright, so if I'm reading from right to left here, I'm going from 3 to 5, cool, I'm following my rules. Notice I'm writing from 5 to 3. I'm going from right to left, I'm adding new nucleotides on right there at the very end there. Okay, I'm still following my rules, so I'm good to go. So I can only read in one direction, I can only write in one direction. Now this strand right here isn't all that hard to make then, right? That was pretty straightforward, we just kind of had to find it and orient ourselves. So this strand is often called the continuous strand, this top strand. Whereas the bottom strand on the other side is, you'll see, we're going to run into some problems. This is called the discontinuous strand. Another name for the continuous strand is the leading strand. Another name for the discontinuous strand is the lagging strand. So we split up our two parent strands, our dark blue strands, into leading and lagging. We have to because of the way we read and write DNA. Alright, so the enzyme that will actually read the parent strand and write the daughter strand that will match up complementary nucleotides, I'm going to draw them in here. I'll draw them in dark red. Of course, he has a name, and you already know his name. His name is DNA polymerase. Alright, his name is DNA polymerase. So if I go back a few slides, all that gobbledygook on the text should hopefully make a little more sense. Okay, DNA is read only the 3 to 5 direction, and it can only be written 5 to 3. And as we'll see in a moment, there's a reason for that. It's because of the 3' OH, the 3' hydroxyl, is what allows us to create the phosphodiester bond in the backbone of DNA. Remember, phosphodiester bonds, backbone of DNA. It's DNA polymerase that actually does the reading and the writing. So this does create an issue because that strand, we can only really easily read and write that top strand. Alright, so here, we're looking at the top strand, we're looking at the leading strand. Really quick, we're going to look at the chemistry again. Notice that 3' OH on the 3' end of that backbone. When we attach this phosphate group right there, we need the OH so we could get out, remember this is a dehydration reaction, so we can get out water. So this is actually the reason why we can only write 5 to 3, we need the 3' OH. We're pulling together a lot of things from the entire quarter here, aren't we? Okay, so because of the way we can only read and write in one direction, again we have to separate our DNA strands into a leading strand and a lagging strand. The leading strand is read 3 to 5. We lay down complementary nucleotides 5 to 3. Pretty straightforward, right? So the strand is also called the continuous strand because once we're on there, we just keep going. The lagging strand, we have to find this weird workaround. Sorry, just hiccup there. The lagging strand we have to read in these little short, weird, backward hops. And we still have to follow our rules. We still have to read, right, 3 to 5, and we still have to write 5 to 3, but as you'll see, it's a weird way that we do it. Okay, so let's go back and let's start writing this down. Let's look at what happens on our lagging strand now. So, again, I have to be reading 3 to 5, so from left to right here, and let me draw a little arrow to signify that. Reading left to right here. Okay. So, in order to do this, what I'm going to do is I'm going to start right at the origin of replication, and I'm going to lay down a chunk of daughter strand, and then I have to hop backwards, lay down another chunk of daughter strand. I'm going to hop backwards, all my enzymes are going to physically move, lay down another chunk of daughter strand. So what I'm doing as an enzyme is I'm first creating this piece of daughter strand, and then hopping backwards and then creating this piece of daughter strand, and then hopping backwards and then creating this piece of daughter strand. So notice, by doing that, I'm still following my rules, right? I'm reading 3 to 5, I'm writing 5 to 3, okay? But, so I'm technically chemically moving in this direction, but I'm still, ultimately as a whole, I'm moving towards the left. So I call this backstitching. I know it's not technically backstitching, but that's what I like to call it. These little backwards hops, these little backwards stitches that we do to circumvent this whole problem of, only being able to read DNA and write DNA in one direction. Alright, so what this looks like on the big scale then, and here we just see one replication fork right there. Okay, these little pink things, we'll come back to them in just a moment, but there's the beginning of new DNA strands. In this image, the bottom is the leading strand, the top is the lagging strand. So notice, as soon as we attach, we can just go forwards with our leading strand. We're good to go, it's continuous, there's no breaks. Our lagging strand we have to attach, and then synthesize, and then a hop backwards, and attach, and then synthesize, and then a hop backwards, and attach, and then synthesize. And as we do that, as our replication fork proceeds to the left here, we are going to glue together the DNA backbone right there. We're going to do some repairs there created by those little hops. Alright, so let's add some more to this image here. We're not done yet. Okay. It's still the same DNA polymerase that's doing all this hopping. So let me draw him in here. Still DNA polymerase. Okay. And here's the thing about DNA polymerase. He's really, really picky. He's like a pop star, right? He needs to have things his exact way before he will start working. So you have to open up the hotel room for him, you gotta lay out all the chocolates on the pillow for him, right? You gotta stock the mini bar. He will only do his job under certain conditions. And one of those conditions is, of course, reading and writing only in one direction. The other condition is that he will not attach to single-stranded DNA. Or single-stranded anything, actually. So when we first open up our replication fork, look, right there, single-stranded. What we have to do to fool DNA polymerase into attaching them, because look, he does eventually attach, is we have to insert a small piece of RNA, green right there. That's a tiny little piece of RNA. Okay. As long as we have that small piece of RNA, we have a tiny little chunk of double-stranded nucleotides right there, and that fools DNA polymerase into attaching, and then starting to do his job, starting to synthesize. So any time that we want to allow DNA polymerase to attach, we have to lay down that little chunk of RNA. We call that little chunk of RNA a primer. We call that little chunk of RNA a primer because it primes DNA polymerase. So let me add my primers in this image now. There's an RNA primer. Every time I reattach, I'm going to have an RNA primer. So I have 3 RNA primers down here on the lagging strand. Let me label these now. RNA primer. Okay. RNA primers. Okay. So on our lagging strand, we have 3 RNA primers, one for each of these weird DNA RNA chunks down there. One final bit of terminology here, okay, on the lagging strand. So each of these chunks, let me draw a box around them. Each of these chunks, let me use a different color. What color do I want to use? Well, let's do yellow. Right here. Each of these chunks has a name, of course. They're named after the scientist who first characterized them, a Japanese scientist. They are called Okazaki fragments. Okay, so in this image we have three Okazaki fragments on the lagging strand. There's no such thing as Okazaki fragments on the leading strand. Now, if I recall correctly, these Okazaki fragments were actually really discovered in the 70s, so pretty recently. So right here, in this image, here's one Okazaki fragment. Here's a second Okazaki fragment. As we proceed forwards, again, we're filling in the backbone, we're removing the remnants of the RNA primer. RNA primers are here in pink. Here's a third Okazaki fragment. Okay, this is just showing again, saying DNA polymerase, don't worry about the 3 here. Okay, he can only bind double-stranded nucleotides, but the problem is we of course start with single-stranded once helicase does his job. So we lay down these little primers so DNA polymerase will actually get in there and start doing his work. And both DNA polymerases on both the leading strand and the lagging strand, we have 2 of them, okay? They will keep going forwards, assuming everything's hunky-dory, until they meet up with another replication bubble that's proceeding in the opposite direction. Alright, a slightly different viewpoint. This is from another textbook. Here's our topoisomerase. Okay. Here's helicase. This is actually kind of what he looks like. He looks like a demented flower. Here's our guy making an RNA primer in yellow. Here's DNA polymerase attaching to the RNA primer, and he's making the leading strand. Don't worry about these things right here. Although I do want to point out they kind of look like peeps, don't they? Alright. On the lagging strand, again, I'm going to come back. Here's our lagging strand, a little bit more complicated. Here's topoisomerase, here's helicase. Here we're laying down the RNA primer. Here's our DNA polymerase, moving backwards though. Here's another RNA primer, and DNA polymerase, after he finishes this first Okazaki fragment, he's going to hop backwards, attach this new RNA primer, and synthesize forwards. We're going to remove the RNA primer, fill in the gap using a couple other enzymes. And here's those couple other enzymes, right there and right there. And DNA polymerase is going to keep doing his job. He's going to keep hopping backwards, attaching to your previous RNA primer, and going forwards. Okay, a little bit about that RNA primer. The RNA primer is about 10 to 15 nucleotides. Okay, and they're laid down every few hundred nucleotides on a lagging strand. And they just, again, they act as little platforms for DNA polymerase to attach to, because he will not attach to single-stranded DNA. And the repeated chunks of RNA and DNA, remember those are named after the Japanese dude, Okazaki. Okay, so at the end here, we end up with one strand that's pretty straightforward, another strand that's a little harder to fix. So let me draw the end result here. Alright, I'm ignoring the multiple replication bubbles for now. RNA primers in red. Okay. Up top, this is our leading strand. On bottom, this is our lagging strand. Remember, on our lagging strand we had to have multiple RNA primers in order to have DNA polymerase come in and attach. Oop. Too far. There we go. That one was my fault. Okay. Alright. So in order to finish this entire process, we of course have to remove those RNA primers. Alright. We only have one to remove on the leading strand, but we have hundreds to remove on the lagging strand. And so let me remove those RNA primers. Nope, okay, we can remove the parent strand too using the eraser, never mind. Let me just redraw this. We're going to remove those RNA primers. We're going to fill in those RNA primers with DNA. But notice I still have these little gaps left over right there. So there's a little gap, there's a little gap, there's a little gap. I can't fill those in very easily, okay? Those are a little bit harder to fill in. So I have to bring in yet one more enzyme in order to fill in those little gaps, and the enzyme is called DNA ligase. So he's going to seal the gaps in the backbone right there. Okay? Now DNA ligase doesn't just seal in the gaps, you know, when during DNA replication. He'll also seal any gaps introduced by, well, any process. So one of the processes that can introduce single-stranded gaps in the backbone of your DNA is actually sunlight. So whenever you walk out in the sun, you are creating thousands of little breaks in your DNA backbone due to UV light, and it's DNA ligase that will repair that. So if you have a defective DNA ligase, well, then you have to stay out of the sunlight at all costs because you can't repair the damage from the sun. This may have actually been the part of the origin story behind vampires, is those people who have defective DNA ligase. Now you will notice one thing, maybe, if you aren't brain dead by now. Looking at, making my drawing a little more accurate, looking at this piece right there, notice this RNA primer is not replaced. That's because there's no previous there's no previous chunk over here for DNA polymerase to fill in and replace that RNA primer. So this little chunk right here just breaks off. It's not used anymore. So every time we replicate our DNA on the lagging strand, we lose a tiny, tiny amount of DNA off the very, very end of the DNA molecule. Okay, I'm going to come back to that in just a moment. Let me show you what this entire mess looks like together. Here's our topoisomerase. Here's our helicase. Primase is the one that lays down the RNA primer. Here's our DNA ligase, right? Here's our DNA polymerases. All of this is just a huge disaster zone, and this is actually close to what it really looks like. That relatively nice, neat drawing that I made for you guys is not quite accurate. Surprise, surprise. Although this version is way easier to comprehend what's going on. With this, well, more true to life, but messier. Okay. Now you may be asking, why are those little primers made of RNA, right? Why not make them out of DNA and not worry so much about replacing them? So here's a potential hypothesis as to why. An enzyme, and that's the RNA primase, the one that lays down the primers, that starts chains anew, that can work just off a single-stranded, cannot be efficient at self-correction. Even if the copies retained in the final product constituted as little as 5% of the total genome, the resulting increase in the overall mutation rate would be enormous. It therefore seems likely that the use of RNA rather than DNA for priming brings a powerful advantage to the cell. The ribonucleotides in the primer automatically mark these sequences as suspect copy to be efficiently removed and replaced. So, due to just some weird chemistry, whenever you lay down nucleotides on a single-stranded piece, you just attach to a single-stranded piece of DNA and you create nucleotides, you can't really double-check your work. But if you work off an already pre-existing double-stranded bit of nucleotides, you can check your work. So, by using RNA, it's really easy to say, well, this part of the genome, this was created by an enzyme that wasn't very accurate. So we have to remove this and replace it. That's what this is saying. So this is the current best hypothesis as to why we use primers made of RNA. Okay. I love these series of images. Right here. I stole this from a professor at University of Washington, so thank you. If you kind of just go back and forth, if you have the PowerPoint slides, whoop, don't do what I did. Don't do that, definitely. There we go. You can really see on here what's happening during DNA replication. Here's the leading strand up top. Here's the lagging strand. There's an invisible RNA primer right there. In red are the RNA primers. Okay. So here we go. Here's an Okazaki fragment. And as the replication fork is going forwards, remember we have our DNA polymerase right there, proceeding this way. We have another DNA polymerase right there, proceeding this way. Okay, here's our topoisomerase and our helicase right there. There's our helicase, the demented flower. Here's our topoisomerase guy. Okay, so we keep going forwards, keep going along to the left here until we reach the end. At this point, we've taken out all of our RNA primers, we remove the RNA primers, and we seal the backbone almost as soon as we make those Okazaki fragments. Notice we're left though with a small bit of RNA primer that we have no ability to fill in because there's no double-stranded nucleotides over here. So that small little bit of RNA primer eventually it leads to this entire chunk of single-stranded DNA being broken off. So we have a problem at the very end of DNA replication. And of course we have a name for that problem. It's called the end replication problem. Alright. And this only happens on the lagging strand because the lagging strand needs those repeated RNA primers in order to be synthesized. We have that leftover RNA primer at the very end that we have no ability to remove and fill in. So this unreplicated end right there, through every round of DNA replication, is broken off and lost. So every time your cells divide, every time you do DNA replication, you lose DNA off the ends of your chromosomes. So what that means then is that we have to have a way to regrow those ends, don't we? Okay, or we have to have a way to protect ourselves. Most of our cells have sort of a protective buffer you can think of just nonsense DNA on the ends. And that nonsense DNA doesn't really code for anything. Its whole purpose is to be eaten away through many rounds of replication. These nonsense DNA buffers, they are called telomeres. And they're just repeated nucleotide sequence repeated over and over and over again hundreds of times. Given enough time, if you live long enough, if your cells divide enough, however, the chromosomes would shorten ultimately into nothing. And this is normal. This is a normal process, right? Especially for our somatic cells, this gives them a natural life limit. But if you think about it, this is not good for all cells. Think about the cells that give rise to your sperm and eggs, right? Every time those guys divide, you don't want their chromosomes to shorten, because that would mean that you pass on shortened chromosomes to your kids, and their kids would pass on even more shortened chromosomes to your grandkids. So we have to have a way to regrow our telomeres in some cases. And of course we have a special enzyme that regrows telomeres. It's called telomerase. Telomerase. So telomerase regrows telomeres, but only in certain cell types. Here in this image we can actually see mitotic chromosomes. These are really cool. In blue, that's regular DNA. The little yellow lights at the end, those are actually telomeres. Alright, so here's telomerase right here. This is the enzyme. And in orange we have the DNA. And the cells that express telomerase a lot are cells that are rapidly dividing. So germ cells, those cells that give rise to sperm and eggs. Stem cells, their whole job is to divide, right? They express telomerase. Your skin cells, remember your skin cells divide very rapidly, express telomerase, although not very much. Cancer cells express telomerase. Cancer cells will keep dividing almost infinitely. Remember when we were talking about the hallmarks of cancer, we said that one of the hallmarks was replicative immortality? Well, here's why. Cancer cells will reactivate telomerase that has gone dormant. Everybody else though, everybody else will just eventually shorten their chromosomes over time. So we think that this may be one of the things that actually leads to aging. So there are many mechanisms that lead to aging, and this is probably just one of them. And the reason why we think this is, well, if you have increased telomerase expression, it's been linked to a decrease in aging. Now you're kind of going, well, cool. Let's express telomerase in everybody, everywhere, we can live forever, right? Well, not quite. Because cancer cells also use telomerase, this is just adding an additional hit. It's enabling one of the hallmarks of cancer. So, old age and cancer both compete. This is also why cancer is so interesting. Okay, so during DNA replication, we do make mistakes, right? In fact, the mistakes made during DNA replication are one of the biggest sources of what are called mutations. So mutations are changes in nucleotides, and despite our huge ability to repair mutations, they still persist. About 70 somatic nucleotide mutations, so this is just in our somatic cells, as one somatic cell divides into another somatic cell, about 70 mutations will arise every time a cell divides. And if you really, really zoom out and you look at, say, a population of people of about 10,000 people who are diploid, that means that every cell has divided so many times, and we've had so many different mutations that every possible nucleotide substitution will have occurred. Every possible mutation will have occurred about 20 times in the course of just 1 million years. So, now we know that most mutations are neutral, right? Of those mutations that are not neutral, the vast majority are harmful. Just a tiny amount are beneficial. So we have to very carefully control the rate of mutation. And in fact, that itself might be controlled by evolution. So for us, our mutation rate is, despite what these numbers might imply, our mutation rate is actually quite low. If you look at viruses, on the other hand, their mutation rate is sky high. In fact, when viruses replicate, they make so many mistakes that they produce offspring, little baby viruses, that 9 times out of 10 can't even survive. Their genes are so severely mutated they don't produce functional proteins. So for many viruses, the progeny failure rate, the chance that their offspring are going to just die right out the gate, is 90%. Now the interesting thing about that, because the mutation rate is so high, there's also a very, very good chance they're going to acquire good mutations, at least good for them. So viruses also evolve very, very quickly. Evolution is simply an accumulation of mutations as well. So, another kind of interesting quote here. The current mutation rate in humans limits the number of essential proteins that organisms can depend on to about 30,000. Any more proteins, more than this, right? And the probability that at least one critical component will suffer a damaging mutation becomes catastrophically high. And that's about as many proteins as we see in humans, about 30,000. Okay, so I'm going to leave this discussion about genomes for another day. Thanks as always for sticking with me. I appreciate it.